all these important constituents which eventually makes us. So those cells has to be some difference between them in the rigidity or in the structural or chemical formation. As we look at them, we have different functions, and if we have different functions, we must have different structures, because structure defines the function, as we have learned. Okay, so that makes the difference. So this, in this picture, we've drawn a animal cell. So this is an animal cell, the picture of an animal cell. So in case of animal cell, we have this cell membrane in the outermost layer, and we in the middle we have protoplasm, we have the nucleus, and the rest of it is cytoplasm. In the cytoplasm, it acts as a matrix, and we have different organelles such as mitochondria. This is called mitochondria, and this is called Golgi bodies. So this is Golgi bodies, and we have this is called lysosomes, and we have, for instance, we have this nucleus, very important one, and we have this, what is it called? It is called cytoskeleton system, and we have small dots like this in this cytoskeleton, or it is called endoplasmic reticulum 2. These dots are called, what are they called? They are called ribosomes, because they are very important to produce proteins, so they are called ribosomes. So we also have this ribosomes. Okay, so now let's talk about all these small organelles and look at their functions in an overview. We don't go to the detail of their function and structure, we just have an overview in this case. So let me erase this first and then discuss about these organelles independently. But before going to that, I must say the cell membrane, we have in the cell membrane, we have this all blue colored things, we call it the protein. So these are membrane proteins. So membrane proteins are very important, because these membrane proteins are actually having different functions, like the entrance or exit for different molecules, such as for instance for different ions to pass through these membranes and all these things. These proteins help them to be transduce a signal which comes, so it helps them to bind with a ligand to make contact with it, or it helps them to recognize a particle, a foreign particle, and to engulf the foreign particle and kill it, and all this type of functionalities. So these proteins over there, they help them to be transported. So membrane transport is derived by these proteins, and all these very important functions are done by these membrane proteins. So cell membrane is based upon the production of phospholipids. We have an arrangement of two layers of phospholipid to make this cell membrane, basic structure of a cell membrane, but we have integrated these proteins, and these proteins actually give them the lots of variations of functions. Okay, so don't forget about these proteins, they are very important. So let's discuss about the functions and the overall features of this organelle, cellular organelles. So we have cellular organelles floating in cytoplasm. For instance, take the first organelles after the cell membrane, we have to take this nucleus. We have talked about nucleus before. This nucleus contains 4 different regions. So if we draw this, if we write this nucleus, can divide it into different regions. We have 4 different regions. We have a nucleolus, we have what is called a nuclear membrane, and then what we have nucleoplasm, and we also have chromosomes. Okay, so this is what a nucleus made up. So nucleus not actually made up, because nucleus have this all these things. So it has a membrane, its own membrane. This is two bilayered, and it has a nucleoplasm. That means it has a matrix too. It has a matrix like cytoplasm. Actually, it is a part of cytoplasm which is taken away by this another type of membrane like nuclear membrane, and it has the chromosomes where the DNA actually resides, and it has a nucleolus. It's a very tightly packed or very dark colored patch found in the nucleus, which is very commonly visible by using the microscope. This is called the nucleolus. So this is a picture of a nucleolus. So what nucleolus does is the production of ribosomes. Ribosomes are very very small protein like structure. They are made up of proteins and rRNAs or structural RNAs, and those RNAs and proteins arrange together to make this organelles like our ribosomes. So ribosomes help or acts like a kitchen for making proteins. So with the help of mRNA and tRNAs, they can arrange themselves to make proteins. So the amino acids are arranged to make proteins using the workbench of this ribosomes. Okay, and we also and so this is the overall structure of nucleus, and in this structure of nucleus we have these chromosomes arrange themselves. They are coiled with each other to form a haphazard structure like this, but when the cell has to divide, we needs to open or unzip this DNAs. So then only the nuclear membrane is degraded and the nucleolus just strips off, and this DNA are come together and make an alignment in the middle of the cell. When we will discuss about the cell division, we will talk about this. Okay, so nucleus is a brain. It is called the brain of a cell. So why it is called the brain of a cell? Because it controls or regulates the functions of different cellular organelles. For instance, it has this DNA, because DNA contains all the necessary information. It has the DNA, so it has those informations. It sends those information via the RNAs, and via these RNAs it can make those proteins. That proteins perform a particular functions, and when those functions are established, we have an enormous amount of change in the cellular functionality and structure. So that's why they are called the brain of a cell, because they control. This is the control room of a whole cell. Okay, so after this nucleus we have to talk about so rest is cytoplasm. In the cytoplasm we need to divide those cytoplasm in small different groups or places. Okay, and that is done by the cytoskeleton or ER. ER means the endoplasmic reticulum. So this structure is called the endoplasmic reticulum. Why an endoplasmic reticulum is important? Because of a very simple idea of physics. If we the matter of creation of this endoplasmic reticulum is to divide the cytoplasm in small places. So dividing the cytoplasm in small places. So why we need to divide cytoplasm in small places? Why it is very important? Now let's look into a very important form of physics. So what is it? Let's talk about it. So for instance, suppose we have a cell here that can work the particular functions so we have some works to be done, and that cell makes those work. It takes 10 hours to do those works, those necessary stuffs. So again we have another cell, and in this cell this cell is divided into groups. It's divided into groups. So when it is divided into groups, it can do the same work in 2 hours. How can it possible? How can it probably be possible? Because what we are looking at is surface to volume ratio. So we have a cellular surface and we have a volume. So we have a surface to volume ratio. If we think about this surface to volume ratio, then you can find out this place. So suppose we have this whole cell, so we have no divisions in the cytoplasm, so we have a whole cell, we have to do works, so we are doing this works. But when you divide these places, we have a various surface area, we have a larger surface area or more surface area. For instance, if we add all those surface areas, we have much more surface area than this one. So we have a surface area of here S1, in this case we have a surface area of S2. So this S2 is further greater than this S1 or the surface area of the previous cell. Why? Because when you divide those things, the surface area is actually increasing. Okay, so what is the necessity of increasing a surface area? Because because all those chemical reactions that is going on throughout the cell is going into a place or a surface. It needs a place to do all this work, because a cell has to do many works. So in this case, many times the cell actually jam with the byproducts and all these things, and then suddenly the cell stops to make function properly. Okay, but in this case we have many surface areas of huge surface area in the same volume, but huge surface area, and that gives the cell more and more place to do work. So it has more and more place to do work. That is important, because we need place is very necessary for doing chemical reactions, and that is why it's very important to make those cytoplasm separated, to make separate chambers that has a speciality to do a different kinds of work, and that framework is provided by this cytoskeleton. The cytoskeleton, we call it the endoplasmic reticulum or ER. So this ER is made up of different proteins like different intermediate filaments, different microtubules, which arrange themselves to make this cytoskeleton. And we not only found this cytoskeleton in a particular place of a cell, this cytoskeleton can be found in throughout the places. And actually not only they are providing the place or separate places for doing a particular job, but they also provide a channel for these organelles to walk through, because these organelles has to be motile, because if the organelle stays in a particular place, then the actions can't be haphazard, because all these things are going on here is about chemicals. So it releases something, it has to take something. So in between this distance Brownian motion can take place, and all this type of works can take place. So ultimately it takes a longer time to reach this substance which is released by this organelle into this portion. Okay, so this is an example of why it is important to the organelles to be motile. So organelles has to be motile. So if the organelle is motile, then the organelle travels itself to the particular place where it needs to be there, and it secretes something which is needed in that place. That's why it's very important. And this cytoskeleton on this endoplasmic reticulums give them the area or the region to walk through it. Because when you look at this detailed structure of endoplasmic reticulum, we can find different molecules, different proteins such as dynein, different proteins such as kinin and actin proteins. So these proteins make the filaments. They are small structural proteins. They arrange themselves to make longer filaments, and those filaments give them a line to walk through. So we have an organelle, suppose here like that, we have an organelle here, that organelle has to be walked through that. We have here a motor protein, that protein is eventually going to walk through this cytoskeleton system, and that's how cellular mobility can be possible, or organelle mobility is possible, and it is very important. Okay, after the cytoskeleton study, what we want to discuss is about, for instance, say Golgi bodies. So what are the action of Golgi bodies? The Golgi bodies are packaging vesicles. We can call it the packaging vesicles. So why are packaging vesicles important? Because we have all those necessary chemicals and equipments to us, and we have to package themselves. So we have to release. Suppose a cell has to release some important parts, so release an important proteins that act as a signal, and that eventually gonna dock into the nearby cell, and that triggers the set of reactions, and that's why we need these Golgi bodies to secrete those those proteins and all these things. So Golgi bodies is very important for secretion purposes. So Golgi bodies actually process all those proteins. So the protein is created into these ribosomes. Using those ribosomes, and those ribosomes are placed on the cytoskeleton, and they walk through the cytoskeleton, and the protein is produced into the throughout with the help of this ribosome, and this protein is traveled into the cytoskeleton in those compartments, and they walk through that compartment, and finally they are connected with these Golgi bodies, because these cytoskeletons are very important. They are connecting all those organelles together, and they are connecting all these organelles together. They are connecting Golgi bodies to it, and mitochondria to it, lysosomes to it, and all those things to it. So ultimately that protein is going to travel through these Golgi bodies, and Golgi body is further process that protein. It attaches some signaling molecule to it, which eventually is going to trigger whenever it docks into the nearby receptor of a cell. So it's very important. So how it works in a nutshell, we have this cytoskeleton here. So you have the endoplasmic reticulum, for instance, like this. We have the Golgi bodies, for instance, like this, and we have a cell membrane in a final layer like this. Okay, so a protein has to be made. We have this ribosomes over there, we have this ribosomes, we have this mRNA, we have tRNAs sitting on it, and they contain those amino acids, and they attach those amino acids together to make a long protein chain. So suppose this is the protein chain, it is made, and this instruction of this whole thing come from this nucleus, and they make a protein, and the protein is eventually going to fold inside this endoplasmic reticulum, and the protein, we have a folded protein, and that protein travels from this endoplasmic reticulum to the Golgi bodies via different vesicles. And those, suppose a vesicle, we have a vesicle like that, we contain the vesicle contains this protein, and vesicle go into, and it eventually going to dock into this Golgi body, and whenever it docks into this Golgi body, it releases this protein to the Golgi bodies, and this protein is further modified. For instance, the signal molecule is added to the protein, and then finally another vesicle docks out from this Golgi body as excision like this, and it's eventually going to travel from this direction, and eventually going to end up with an exocytosis process. Exocytosis is a process of releasing cellular material. So we have this protein with the signaling molecule, which is processed by Golgi bodies, and finally this protein is going to release from the cell. Okay, so this system is called GERL system. Okay. It's a very important system. So this is a secretion, a normal common secretion system in cell, and in this example we can find an enormous effect of cellular coordination. So we have lots of different organelles. For instance, we have nucleus, which contains the DNA, which eventually gives a signal. We have ribosomes, which sit on those endoplasmic reticulum, that actually process that protein, that actually make that protein using mRNA, tRNAs, and amino acids. And we have Golgi bodies, which process that protein at those signal sequences which is needed to be released, and then we have plasma membrane, which actually helps to release that protein to the outside. So we have a coordination. So cell means coordination. So we need coordination to work. So cell always work in coordinate manner. Without this coordination, the cellular system will collapse. So it's very important to have these coordinations. Okay, we have a structure, we have the function of ribosomes here. So ribosome in this structure, for instance, say we make this ribosome two structure units. So ribosome is made up with these two structures. So it is made up with two subunits. What are those subunits are called? The subunits are called a big subunit, another is a small subunit. And not only they have the big and small subunit division, but they also have three sites where the tRNAs can sit. Okay, where the tRNAs can sit, and they have a terminal portion through which an mRNA can pass through. Okay, so they are a scanning, they have made in such a way like a scanner. So they can scan those mRNAs. The mRNAs contains codons, the sequence for amino acids, and to read that codons, tRNAs comes in to reading that codons and having those amino acids attached to themselves, and they sit on this tRNA attaching portions of ribosome, and they transfer the peptide bonds and they make a long protein chain. That's how ribosome works. Okay, after the ribosome we must talk about these red blobs, we call it the mitochondria. And what is an action of this mitochondria? Because mitochondria is business. Mitochondria means economy, it means the currency, because mitochondria means ATP. I must say mitochondria means ATP, or the energy currency of the cells. So cells need energy, not only cells, we need energy, means the cells